Hello students, in previous section we have studied about nucleic acid DNA and RNA. In this section we will study on enzymes, the another biomolecules enzymes. Biomolecules, enzymes. Enzymes are also biomolecules. How the enzymes were discovered? Firstly, we will show that enzyme was discovered by whom? Firstly, the enzyme was discovered by Louis Pasteur. While Louis Pasteur performing its fermentation process, then he saw that there are some substances, some microbes which increase the rate of reaction. Those substances he called ferments, not enzyme, ferments. Louis Pasteur, while he performing fermentation process, then he saw that there are some substances which affect the rate of reaction. He called those substances ferments. According to Louis Pasteur, ferments nowadays are enzyme. Then another scientist called Buchner, he isolated the chemical substance from the yeast. Isolated the chemical substances from yeast. He called these isolated chemical substance Enzymes complex. Enzymes complex. But later the name enzyme was proposed by Kohane. The term enzyme was proposed by Kohane. So we can say that there are the three scientists' role to find out the enzymes. The enzyme word which was given by Kohane, and Buchner proposed that Enzymes complex, and Louis Pasteur ferments. Later it was seen that these chemical substances enhance the rate of reaction. It means they increase the rate of reaction. But there are some other substances, some chemical substances which also reduce. So the definition of enzyme is these are the chemical substances. Definition of enzyme is the chemical substances which alter the rate of reaction. Firstly, it was assumed that all these chemical substances only enhance the rate of reaction, but there are some reducing chemical substances also. So we can say that the definition of enzyme is the chemical substances which alter the rate of reaction. How we can show that the enzymes play very important role? Suppose with the help of an example, CO2, it combines with water and makes carbonic acid, H2CO3, in absence of an enzyme. If this process occurred in absence of an enzyme, then the product will produce carbonic acid, but the production rate is very low. The production is nearly 200 per hour, or we can say that per 60 minutes, or we can per hour. If carbon dioxide react with water, it produces carbonic acid, but the production rate is very low. It means 200 product per hour. But when we perform this activity, perform this chemical reaction in presence of an enzyme, the chemical reaction becomes fastest. CO2 react with water in presence of an enzyme, and that enzyme is carbonic anhydrase. Carbonic anhydrase. In presence of this enzyme, the product carbonic acid is produced, but the production rate is very high. It means 6 lakhs per second. In both the chemical reaction, the product is carbonic acid, but the production rate in presence of an enzyme carbonic anhydrase, the rate of reaction becomes very fastest. So we can say that these enzymes have to increase the rate of reaction. This is the first example of the use of chemical enzyme. The another one is, we can take another example. For example, there are some substrate. This one, like these are some substrate, and they carried some energy, such like as it carried 2, 3, 4, 5, 6, 7, 8, 9, 8, 7 or 10. These are the substrate which carry energy. They carry activation energy. These are the substrate. They can change into a product. While they change into a product, they need activation energy is 5. If there is 5 activation energy, they can change into a product. Then we can see that only those substrate able to change in product which have the energy nearly 5 or more than 5, not less than 5. The substrate are not able to produce product. So we can say that those substrate only which have energy 5 or more than, it means 1, 2, 3, 4, 5, 6, 7, 8. Only 8 products may be produced in absence of an enzyme. These are the substrate which carry the activation energy. While they change into product, they need energy, and at that time activation energy required 5. So we can say that only those substances change into product which have energy 5 or more than 5, but less the rest substances are not able to change into product in absence of an enzyme. But when we do perform this activity in presence of an enzyme, we can see that these enzymes are able to change all the substrate and change into the product. For example, when we use an enzyme, enzymes have some depressions. Enzymes have some depressions. This is an enzyme. These enzymes have this depression, and in these depressions, those substrates which have lower energy than the activation energy, they combine here. This enzyme reduce the activation energy. The activation energy required 5 while the substrate change into product. But when we use enzyme, the substrate which have a lower energy than the activation, those substrate combined with the enzyme, at that time enzyme reduce the activation energy, and all these substrates are able to change into product. It means all substrate 4, 4, 8 and 11 substrate change into product. So we can say that the production rate is high, as well as there is required some less energy than the activation. We can perform this by a graphical representation. For example, this is the potential energy representation by y-axis, and x-axis we can represent by time, potential energy, which is required by the substrate to change into product. Suppose this is the saturation line. The substrate which able to change, which required to change into product, they need some energy, activation energy, and that state is called transition state. This is the transition state. It means the substrate when changed into product. This energy is required. This amount of energy is required while there is no enzyme is used. This is the energy. The energy required in absence of enzyme. But when we use some enzyme, the substrate required lower energy, because we know that, and this is the energy, this is the transition state in which energy is required. While we use some enzyme, the energy required in presence of enzyme, this energy required. So we can say that enzymes are those chemical substances which alter the chemical reaction, rate of chemical reaction, as well as they reduce the activation energy. Enzymes are also able to reduce the chemical, reduce the activation energy. So we can say that enzymes are those chemical substances which alter the rate of reaction, as well as they are able to reduce the activation energy so that all the substrate can change into product. A complex is produced. For example, this one. If this enzyme, when the substrate attached combined with the enzyme, this formed a complex. This is called a complex. The enzyme with substrate. This is called enzyme plus substrate complex. While substrate attached with enzyme, it forms enzyme substrate complex. Clear? Now we can see that the enzymes show two mode or two types of mechanism which was proposed by different scientists. The mode of enzymes, or we can say that mechanism of enzymes of action. How the enzymes perform action? There are two modes. Enzymes mode, or we can say the mode of enzyme, modes, or we can say the mechanism of enzyme for action. It means there are different modes, different mechanisms by which enzymes show actions. How these modes? There are two modes. One mode is lock and key. Lock and key mechanism. This lock and key mechanism was proposed by Fisher. It was proposed by Fisher. How this lock and key mechanism action can be shown by the enzyme. Suppose this is an enzyme. This is an enzyme. There are some depression present in enzyme. These are the depressions. These depressions help into combine the substrate and formation of bond. How these can be performed? This is an enzyme. While this enzyme combined with the substrate, suppose there are other substrate. One is this one, like one substrate is this one, is substrate S1, substrate 1, and another substrate is this one, like This is the S2. These are different substrate. When these substrate combined with this enzyme, because enzyme contains some depression, and in these depressions the substrate are trapped and they combined form a bond. While they form a bond, they change it to product. Then enzyme is released. For example, after this, the enzyme this one enzyme, and it combined with the substrate here. This is the S1 substrate 1 and another substrate 2. S2. What is the role of an enzyme? This enzyme trap the substrate and form a bond between the substrate. As the bond formation can take place, the enzyme free this substrate, and this structure is called enzyme substrate complex. This is enzyme substrate complex. Enzyme substrate complex combine both the substrate or many substrate. They form a bond and release this substrate. So this substrate is released S1 and this is the S2 by forming of bond and it change into product. The enzyme is free. This enzyme is then again changed into another substrate and form a new product. This is called lock and key mechanism, which was proposed by Fischer. One more action which was represented by an enzyme to perform some activity, or we can say that change into product, that is induced fit mechanism. Another mode is induced fit mechanism or induced fit mode. This was proposed by Kusland. He showed that the enzymes are able to form or change the substrate into product only some activation site or active site. So induced fit mechanism function on the base of active site, and there are two active site. One is catalytic active site, and other one is buttressing site. How these site activate our functions? Catalytic active site. This is the site which break down the old bond and formation new bond. This site break down the old bond and make new bond. That site is called catalytic active site. And second one is buttressing site. This site is only the site where the substrate are attached, or we can say that it is the site where the attachment of substrate can take place. The site where the substrate attached. How these sites are performed in an enzyme? Suppose this is an enzyme this is an enzyme. This enzyme contains two sites. One is a catalytic active site and the other one is buttressing site. This is called catalytic active site and this one is buttressing site. As we have seen that buttressing site only the site where the only substrate are attached. These are some substrate. Suppose these substrate like this one. These are the substrate. These substrates firstly attach with the buttressing site. For example, this one. The substrate are attached here with it. This is the buttressing site. Here the substrate are attached. At this position we can say that this is enzyme substrate complex. Enzyme substrate complex. And this is the activation site. This site helps to break down the old bond and formation of new bond. If there are some more substrate rather than this one, if there are more substrate, then enzyme from catalytic site overhead, or we can say that it enhance its site. For example, this one. And it binds with the substrate. This site may enhance and come down to overhead or overlap this substrate, and at this time they form a new bond formation here. The new bond is formation here by the effect of catalytic bond. As the bond formation can take place, this substrate is able to change into product. While this substrate is able to change into product, the enzyme free this substrate and enzyme is ready for another process. And this enzyme is ready for another process, or we can say that the enzyme is ready to combine with the other substrate. So production of product can be take place with the help of an enzyme. In next section we will discuss on some chemical substances which form enzymes and factors which affect the enzymes.